Hello, and welcome back to Introduction to Genetics and Evolution. I decided to add this extra short video because several people expressed that they didn't know a little bit about the scales of organization of genetic data. Now we talk about genes and we talk about how they affect a phenotype, and we talk about how they're hereditary. We mentioned DNA, but we never really said anything about how is it that the DNA sequence encoded in genes actually affects phenotypes. That could easily be the topic for an entire course. In fact, it is a topic for many courses. What I'll do is introduce just some very basic concepts just to help you understand the context of the class that we're doing right now on genetics and evolution. Now, how is genetic information translated to phenotype? Now, we've already talked about how DNA is the primary hereditary material. That is the material that is organized in chromosomes, that is passed from parents to offspring, that is allowing this mechanism for heredity. Now, there are sections within chromosomes that have specific instructions, and these sections are referred to as genes. This is a term that you hear all the time. Now, the question I'm posing to you now is what or how does this impact the phenotype? How is it the DNA sequence that's present in these different genes actually changes your hair color, or your eye color, or your personality, or your IQ? Well, a lot of this, not all of it, a lot of this is done through protein. Now, you hear the term proteins all the time in the context of your diet. Are you getting enough protein? Oh, this kind of food is high in protein. And in fact, proteins are vitally important to many, many aspects of your health, well-being, and development. Some proteins are structural. They're basically putting you together. Some are mechanical. They're essential proteins in various of your muscles, for example. Some of them are biochemical. The enzymes that you use for digesting your food, for example, are largely protein-based. And they can even affect things like cell signaling. So your hormones, for example, have protein components. Now, the central dogma of molecular biology, this is a fundamental principle. And this is an oversimplification, I should stress ahead of time, but it is nonetheless a fundamental principle of molecular biology, is that there are three primary levels of genetic information. They are also interrelated to each other. Now, DNA, as we've talked about, is that hereditary material. It is passed to offspring, but it also replicates itself, both inside your body and in the context of transmitting this genetic information to offspring. So DNA replicates itself. DNA also produces RNAs, and we'll focus on one particular kind of RNA here, referred to as messenger RNA. Messenger RNA is an intermediate between DNA and proteins. There are some forms of RNA that are directly functional, but again, we'll be emphasizing it in this video, the messenger RNA, those ones which are actually putting together the protein sequence. And finally, these messenger RNAs are translated into protein. Now, the structure and content of these proteins are what directly impacts your phenotype, or how you look, how you feel, how you act. Now, proteins are actually composed of chains of amino acids. Again, you've probably heard the term amino acids in the context of things you want to get particular kinds of in your diet. But if you look over here on the right, we have a string of amino acids. So this would be part of a protein. If we zoom in on some of them, we see particular amino acids that you may have heard of: phenylalanine, leucine, serine, etc. Now, how is it we get from DNA to these proteins, aside from having this RNA intermediate? Let's talk about each step just very briefly. So the first step is DNA forming this RNA, specifically this messenger RNA. Well, the formation of messenger RNA from DNA is referred to as transcription. Okay? Now, as you may remember from other courses, DNA is double-stranded, and you have this complementary pairing. So for DNA to DNA, you always have these four different types: A, C, G, and T. These are the four different nucleotides of DNA. A always binds with T, C always binds with G, G always binds to C, and T always binds to A. And you see that over here. So here's DNA to DNA. A to T, A to T, T to A, C to G, G to C. Okay? Now, when you're producing the messenger RNA, you have the same sort of complementary relationship, except in this case, the T is replaced with a U. So this is a uracil. So in addition to being a different sort of compound, the DNA is deoxyribonucleic acid. RNA is just ribonucleic acid. It doesn't have the deoxy facet to it. You see it has the same sort of complementary pairing. So we have a double-stranded DNA that has been separated out, and we have this RNA transcript that is being produced in the middle, and you have the same sort of relationship of nucleotides. So you're essentially making an a copy of the DNA as RNA. You note the sequence here of this RNA transcript exactly matches the one here at the bottom, aside from this switch from T to U. See, TAC, TGCC, UAC, UGCC, etc. So this is how the DNA is making its copy as messenger RNA. Now what happens is this RNA copy of the gene sequence will leave the cell, leave the cell nucleus. It'll enter the cytoplasm, and that's where it directs the formation of these amino acids. So here, this messenger RNA, again, when it comes out, has to produce these amino acid chains. The term for this translation from messenger RNA to amino acid chains is translation. Essentially, we're taking this messenger RNA sequence and we're making it into a very specific string of amino acids. It's not just any amino acids, but it's very specific. And again, we have a code, just like the code we saw before. Now, this code for translation uses 3 nucleotides at a time, or 3 bases at a time. So this is a triplet code. So what happens, this is looking at the messenger RNA here on the bottom. We have an AUG, that is associated with one particular amino acid, methionine. ACG is associated with another amino acid. GUA is associated with another amino acid, etc. So it's going in this triplet code, 3 nucleotides at a time from messenger RNA to the resulting string of amino acids. Okay? This will continue until it gets to what's referred to as a stop codon. There are 3 different stop codons. This is one of them, UAA. UAA is not associated with amino acid, and it terminates the production of this string of amino acids. So we'll continue adding these on until you get to a stop codon, and then boom, done. We have completed the process. So let me show you how you actually identify these. There are tables, these are referred to as codon tables, that show you what AUG stands for, what UAA stands for in terms of amino acids. And here's a standard genetic code table. Here's AUG, as you saw before, is methionine. That is correct according to this table. UAA was the stop codon that you saw before as well. But you see all the different combinations. There's 64 different combinations because it's 4, since there's 4 nucleotides, to the 3rd power, because it's a triplet code. So 4 to the 3rd is 64, and that's the size of this table. There's 64 possibilities here, of which 61 are associated with amino acids. 3 of them are not. There's 3 stop codons that are not associated with amino acid. But we can immediately translate. CCC will give you a proline. An AAA in the messenger RNA will give you a lysine in the amino acid. So we have this perfect genetic code for putting these things together. Now, for closing, let me introduce some areas of the genome, because I'll be using these terms occasionally and I want you to be familiar with them. Again, as we mentioned before, genes are these areas that have amino acid coding segments. Not all of the gene, however, codes for an amino acid. There are some segments of genes that don't. And one example of a segment of a gene that does not do so is the intron. Now, introns are areas within genes that are actually spliced out of the messenger RNA. So an RNA is produced from a large segment of the gene, but then these intron regions just essentially get cut out of the final messenger RNA as it's put together. As a result of that, they do not affect the final amino acid sequence. Okay? That's a very important point. So intron sequences do not affect the final amino acid sequence. The other region I want to introduce to you is what's referred to as intergenic regions. These are the areas that are found between genes. Now, just because it's not found in a protein-coding gene does not mean it's not important, does not mean it does not have a function. In fact, a lot of these intergenic regions are functional. Several of them regulate how much messenger RNA is produced, especially from neighboring genes, but not exclusively. And sometimes these intergenic regions even produce other kinds of RNAs that we haven't talked about here. So let me give you a fictitious example. These are 3 real genes, but I've diagrammed them, not how they truly are. So let's say there's the CHEK2 gene. CHEK2 you may have heard of because there's mutations in it that are associated with a 5-fold greater risk of breast cancer. I have another gene here referred to as NF2, another gene here referred to as SOX3. I've depicted them again fictitiously, so they're not actually right next to each other like this. So CHEK2, as I've depicted here, would have this whole segment which codes for a messenger RNA, and ultimately then is related to an amino acid sequence. There may be a segment here, this is the intergenic region, the area between it. This segment right here may actually affect, depending on the DNA sequence that's found here, may affect how much of this CHEK2 messenger RNA is actually produced. So this segment is actually vitally important, and it affects how much of the messenger RNA is produced, which will then also potentially affect how much of the protein is ultimately produced. So it does have a function, but its sequence does not affect the amino acid sequence of this CHEK2 protein. NF2, as I've depicted here, has two large regions that are associated with amino acid sequence, but also has an intron. So the messenger RNA that's ultimately produced from NF2 in this fictitious example would have this sequence attached to this sequence, but the middle part would not be present. Even though the middle part is there in the gene itself, it is not found in the messenger RNA. SOX10, similarly, has this large protein coding segment, this protein coding segment, this protein coding segment, but it has two introns. Again, those two introns would not be found in the final protein. And again, both of these may also have other sections here in the intergenic DNA that are regulating how much of the messenger RNA is produced of those two genes as well. Well, this was a whirlwind tour, but I just wanted to introduce some of these terms to you. I wanted you to understand the triplet code. I wanted you to understand the central dogma, this idea that DNA makes messenger RNA, which affects the amino acid sequence of proteins. And I want you to notice that the genes have these particular areas referred to as introns that do not affect the final amino acid sequence of protein. I hope that was helpful, and I hope it was interesting. Thank you for joining.